Hey guys, welcome to Pre-AP Biology Cell Cycle 1.5 Part 1. And here we go. Well, we're going to be talking about why cells divide. And first of all, why do they need to divide? Well, the picture that you're looking at right now is a cancer cell, and it's a scanning electron micrograph of a cancer cell. It's a pretty cool picture right there. And it's dividing just because it wants to, and there's no controls on it, and that's what makes it a cancer cell. Cells, however, cells in your body need to divide for one of three reasons. One, growth. Remember, you started off as a single cell, as a fertilized egg, and you now consist of trillions of cells. How did you get that way? Through the process of mitosis, of one cell becoming two, two becoming four, four becoming eight, and so on and so on and so on. Okay. Another reason cells to divide is to repair themselves or repair an organism. If you cut yourself, the cells become stimulated to basically repair the cut that you did and end up filling that cut with scar tissue and whatnot. Also to replace damaged cells. Remember, your digestive tract is subjected to all sorts of abuse from the food that you eat and the stomach acids and things. And the cells lining your digestive tract pretty much are replaced every 2 to 3 days because they become damaged. And the third reason cells divide is for reproduction. And that's a form of asexual reproduction where there's not two parents, it's just one parent. And as it's obvious in a single-celled organism, when that cell divides, it basically creates a new organism. In multicellular organisms, it's not so obvious, but it happens in animals and things like little hydra. They'll actually grow another organism out of part of their body and it buds off. Plants do this all the time. They grow new plants and strawberries are a great example. Strawberry plant goes along and the runner comes out, it goes into the ground, that puts new roots into there, and you basically have a genetic clone in a new plant. So this happens all the time in the plant world. So those are the three reasons: growth, repair, and reproduction. Okay. Here is a picture of all of the chromosomes in a human. We have 46 chromosomes, and this is what we call a human karyotype. And you can see this is the karyotype from a male. How do I know it's a male? Because they have X and Y chromosomes. If this was a female, there'd be two X chromosomes here. So we have 22 pairs. There's one pair, there's two pairs, pair number three, pair number four, and so on. We have 22 pairs of what we call the autosomes. Okay, these are the non-sex chromosomes, autosomes, and one pair of sex chromosomes. Soma means body, and these have been dyed with or actually they've been tagged with a fluorescent protein that we've isolated from jellyfish. And they don't normally look like this, of course. They normally look a little bit more like this. And then we basically would cut these out and then we'd line them up to look like this to make a karyotype. We do this sometimes to see if there's any gross genetic abnormalities, like missing a chromosome, or you have extra sets of chromosomes. And some of you guys may have heard of trisomy 21 or Down syndrome. That's when individuals have three copies of chromosome number 21. So if you look at number 21 down there on the bottom center, instead of just being two chromosomes there would actually be three chromosomes. Okay, this is a human. This is what we call the human genome. The genome is basically the entirety of DNA with an organism has, and you would have these 46 chromosomes in every single one of your somatic or body cells. And these are the cells. These would be your fingernail cells, your skin cells, your hair cells, your liver cells, your heart cells, your cells in your big toe, and every muscle cells, nerve cells, everything. The only cells that would not have 46 chromosomes would be your sperm cells if you're a male and your egg cells if you are a female. In those cells, you would only have half the number of chromosomes, 23 in humans' case, and that would be a haploid cell, Haploid. It means half the sets of chromosomes. We, all of our other cells, we have two sets, or we are diploid in there. So there's a lot of cells. Okay, and it can be in two different forms. It's either in the chromatin form or the chromosome form. In 99% of your cells, it's going to be in the chromatin form, and in a smaller percentage, maybe 90% of your cells will be the chromatin form, and a smaller percentage will be in the chromosome form. What's going on? Well, in the chromatin form, it's uncoiled DNA, and your cell is doing its normal job at that point. All the genes are readily accessible. When your cell gets the signal to divide in mitosis, then that chromatin basically condenses into chromosomes. And it just makes it easier to pass that DNA around, and I'll show you a little demonstration in class of what I mean by that. Um, Generally, your DNA in your cells is going to be in the chromatin, what I like to call the working form of DNA. And then when that cell is getting ready to divide, it goes into the chromosome form of that DNA. And as it's noted in your notes there, you have about 7 feet of DNA inside of every single one of your cells. That's a tremendous amount. How do we get it in there? Well, we do it by coiling the DNA up on these little protein spools, kind of like thread on a spool, wooden spool. If anybody in your family sews, you know what I'm talking about. But these proteins are like little spools, and the DNA is wrapped up around them, and then they wrap up even tighter and tighter and tighter until eventually we get the chromosome. And it's just a way of keeping all that DNA up in a nice, controlled, tight package so it's not broken or damaged when the cell divides. So, some other terminology here. We start off with our DNA. We have basically 146 chromosomes, and when the cell gets the signal to divide, every one of those 46 chromosomes has to replicate itself. Okay, because it has to make two identical cells, so we have to double all the DNA in the cell. And so let's just look at one chromosome in this picture up here. We start with this single chromosome up on top. It gets the signal to divide, and so it replicates itself in a process we call DNA replication. And it makes up a identical chromosome right next to it, okay? And it's attached in this little region we call the centromere. And as long as these two chromosomes are attached, they are known as sister chromatids. Tids means part of, so these are part of the chromosome. Each is one half of the chromosome, although each one of these sister chromatids has a complete set of information that this particular chromosome has. It's just they're identical copies of each other. And then when the cell begins to divide, these sister chromatids are pulled apart, and they're known as chromosomes again. And they'll end up with one copy of each of these sister chromatids or chromosomes now in the new daughter cells. This will be in one cell, this will be in another cell, along with 45 other chromosomes if this is a human, and this one will be in another cell with 45 other chromosomes again if it's a human. So, difference between chromosome, sister chromatids are attached, and when they split up, they're back into chromosomes again. Now we're going to talk a little bit about the control of the cell cycle. And this little diagram is kind of cool. We start off with the cell in metaphase, which is the middle part of the mitotic phase of the cell cycle. And the DNA starts to pull apart in what we call anaphase, and we start getting into telophase here. Then we start to see the cell membrane starts to pinch in something we call cytokinesis, until we eventually end up with two complete daughter cells. So we start off with our parent cell, and we end up with two identical daughter cells down here. Brand new cell. and there's just the normal amount of DNA in the cell, and it's doing its thing until it gets a signal, hey, time to divide. Then the DNA replicates itself during the S phase or the synthesis phase, and the DNA becomes doubled at this point. Okay, so we go from the normal amount of DNA to twice the amount of DNA. And then when the DNA has completely copied itself, we move into the G2 or GAT2 phase. And this is in preparation for cell division. The organelles in the cell, the Golgi apparatus, the endoplasmic reticulum, mitochondria and things start to increase in number. The cell grows a little bit more. And then we'll move into the mitotic phase, which is going to be the division of the nucleus, and eventually we'll move into cytokinesis, which is the division of the cytoplasm, and we'll end up with the DNA is in the chromatin form, and that's what this blue stuff is in the center of the DNA. We don't see any chromosomes. It's in the chromatin form. This yellow is the cytoskeleton. Okay, so this is the nucleus. DNA is in the chromatin form. When the cell gets that, and of course, if this is the S phase, the DNA is starting to replicate. So this is beginning of mitosis. We start off in interphase. The DNA replicates. It's in the chromatin form. We start moving into prophase, and we can start to see the chromatin condensing into chromosomes. By the end of prophase, we have some nicely identified chromosomes. We can see the centrioles out here organizing organizing the mitotic spindle. Okay, which is what will physically move the chromosome. Here's a better picture of it. A really cool picture. Here's the centrioles, and these microtubules are attached to the centromere on the chromosomes, and they're physically moving these chromosomes. The cell has will have a diploid number of 4 chromosomes. There's going to be 4 chromosomes in this cell and 4 chromosomes in this cell by the time we get through the end of telophase. Here's a nice picture of a cell in metaphase. All the chromosomes are lined up in the middle of early anaphase. They're starting to get, the sister chromatids are starting to get pulled. And there we are in telophase. We see they're very well pulled apart at this point. We start to see cytokinesis beginning and things of that. So how do they actually move around? Well, there's these little motors that are found on the centromere of each sister chromatid. And it's a thing called the kinetochore, and on the inside is a little motor protein that physically walks up the microtubule. How does it do that? Well, it's getting its energy from ATP, adenosine triphosphate. And remember, adenosine triphosphate is what the mitochondria make. Well, how do they make it? Well, they take the energy from the food that you eat, in the carbohydrates, the proteins, and the fats, and they turn that, the mitochondria turn that energy and those molecules into ATP, adenosine triphosphate, which is a molecule that all living organisms use to power all their cellular processes. Now, I'll give you a couple minutes. Let's give you 10, not a couple minutes, but give you 10 seconds. See if you can identify some phases in this particular onion root tip, a plant. There's 10 seconds. I was clicking my teeth to count those 10 seconds off. All right, so what you have here, let's see, did you identify any of them? Well, I can see a few here. It's not, most of them are in interphase, and that's what most of the cells are going to be. But here, this looks like, it could be a combination of prophase, maybe late prophase, early metaphase. Here we definitely see anaphase going on. This looks like late metaphase or early anaphase. Prophase, interphase, mostly interphase. Oh, there's a cool cell. This one is late telophase. We see cytokinesis. This in plants, a cell plate actually grows there, but we can still see some chromosomes in there. Okay, so that's kind of cool over there. Anyways, moving to grow a cell wall between the nuclei in telophase, late cytokinesis, where the animal cells just kind of just pinch in on themselves. What's doing that? The microtubules are pulling in on the cell. Bacteria are simple. I'm not going to say much about it, but they reproduce via a process called binary fission. Basically, the DNA just copies itself, starts at one end and copies itself until we get two copies of the DNA. No, the DNA is in the form of a single circular chromosome. We don't have linear chromosomes here. It happens very quickly. There's not much DNA in a bacterial cell. It takes anywhere from 5 to 10 minutes. Whereas in a human cell, it'll take upwards of 24 hours. So bacterial cells reproduce very, very, very quickly. And why? If you get a bacterial illness, you can get sick really quick. Well, I think that's going to bring us to the end of part 1, part 2, and we're going to go into part 3, talking about cancer cells a little bit. What the heck is cancer? Well, cancer is just basically cells gone bad. And here we start off with comparing normal cell division and cancer cell division. We got a normal cell up on top, and then on the left-hand side, those cells will divide normally until they reach the maximum number of cells that can fit in that spot, and they stop. Where cancer cells, which have damaged mutations in their DNA, actually generally there are going to be mutations in the proteins that regulate when a cell stops dividing or when it starts to dividing. So we get problems in those genes, and then we get cancer. And basically, there's no control, and the cells just keep on growing and growing and growing until they make a big mass called a tumor. And cancer, as you well know, mostly occurs in people that are older. Why? Because we accumulate mutations as we age. The older we get, the more mutations our cells have. So therefore, cancer is primarily a disease of the aged. Okay, yes, kids do get cancer, and that's just tough luck. I mean, it's bad. It's unlucky. It's a bad chance. It's just random. It's random that this happens, and they just randomly had a mutation in that particular cell, in the right spot. But generally, this doesn't happen. And again, cancer is a disease that is more pronounced the older you get because you accumulate those mutations. So, going back to our cell cycle that we saw earlier, when we have the G1, the S, the G2, and the M phase. Well, before the cell actually moves on to these phases, there's a little checkpoint. And there's the G1 checkpoint, and that's just saying that the cell is grown, it's big enough, it's ready to replicate itself. And if everything's okay at the G1 checkpoint and the cell receives a signal, it will move into the S phase where the DNA is duplicated, and then the DNA will replicate itself, and then we move into the G2 phase. But before we actually move into the M phase or the mitotic phase, there's another checkpoint to make sure that the DNA has been copied correctly, there's enough cell organelles, the cell's the right size, and so on, so on, so on. And then we begin mitosis. And then there's another checkpoint, the M checkpoint, or the metaphase checkpoint, which checks to see that the cells are all lined up on the cell plate correctly, that they're attached, the centromeres are attached to the microtubules, so that during anaphase, when the sister chromatids are pulled apart, we're going to get an even division. We're going to end up with, in humans, we're going to end up with 46 chromosomes on one side of the cell and 46 chromosomes on the other side of the cell. Sometimes there's mistakes, so we end up with more or less chromosomes on either side of that cell. Okay, and that can cause some problems. Cancer is also what we like to say, it's density independent. It doesn't, basically, if there's too many cells there, cancer doesn't care. It just keeps on growing. Most cells, as you can see in this top diagram on the left-hand side, we start off with this is a tissue culture. We throw some cells in this little tray that we grow cells in, and they will start to grow until they reach the edge of that tray. Once they reach the edge of that tray, they stop growing. That's called density-dependent inhibition. And when we remove some of those cells, or like when you injure yourself, they get the signal to divide again, and they fill up the wound or the little gap there, and then they stop dividing again. Cancer cells don't do that. They just keep on dividing and dividing and dividing and dividing and dividing, and that's what a tumor is. It's basically a cell that has lost its inhibition from dividing. Okay. Not only that, as you all know, cells have to get food. How do they get their food? They get it through capillaries and small blood vessels and things of that nature. Well, as a tumor starts to grow, it starts to get real big, and some tumors can get up to 20, 30, 40, even 100 pounds big. They can get big. Well, they have to feed themselves. So what do? They actually send out signals that cause blood vessels to grow into the tumor to feed the cancer. Now, that sounds kind of nasty, but it's pretty darn cool too. And how's it doing that? It's doing it all through cell signaling like we talked about. So here's an example of a benign tumor on the back of a boy's neck, and benign just means it's not spreading. Okay, we can go in surgically and remove this and no problem whatsoever. Tumor, on the other hand, is one that has escaped the confines of its little area, and it's now spreading throughout the body. It's invasive. Okay, it's moving throughout the body, and it's putting its cancer cells all throughout the body, and tumors start to grow in places that are inappropriate, like near your heart, in your brain, in places that start to basically impinge on your organs' abilities to function. And you might start off as painful in one area, or if it's in the brain, then loss of memory or some other things of that nature can happen. So that's a malignant tumor. Benign tumors, not that big of a deal unless it's in a spot that's hard to remove, but they can usually be removed pretty safely. Anyways, that's kind of cancer in a nutshell. It's pretty quick. It's just cells gone bad. And we'll be talking more about cancer biology. It's really cool stuff. And again, when I was your age, when somebody got diagnosed with cancer, that was pretty much a death sentence. But nowadays, it's still a scary word, but most of the cancers that were deadly when I was your age are now curable, and you know we are very, very close to curing many, many, many more. So good things on the horizon that way.